Good morning, children. We were learning about translation. Sorry, we were learning about transcription. And you know that transcription is the process by which the genetic information is copied from DNA to RNA. About the transcription unit we learned and how the transcription process is taking place, the different steps involved in transcription also we learned. And once transcription is completed, what do you call the RNA obtained? Once immediately after the transcription process, the messenger RNA, I told you it is a process by which genetic information is copied from DNA to RNA. So what do you call that RNA formed immediately after transcription? Evelyn?
Messenger RNA.
Immediately after that, you call it as messenger RNA?
HNRNA.
HNRNA, heterogeneous nuclear RNA, or it's a primary transcript. This HNRNA should undergo some processes before it can change into a messenger RNA. Which are those processes, Michael?
And splicing.
Splicing. Two more.
Capping, tailing, and splicing.
Capping, tailing. Okay, splicing, capping, and tailing. Splicing is a process by which introns are removed and exons are joined together. And then at the 5' end and at the 3' end, we add certain substances to provide them protection from degradation. So what do we add at the 5' end, Gayatri?
Methyl guanosine triphosphate.
And at the 3' end, Nandana?
Adenylate residues.
Adenylate residues, polyadenylate residues are added so that capping is also done. Once capping, tailing, and also splicing is completed, HNRNA will get transformed into messenger RNA. Before we proceed to the next process, that is translation, you're going to learn about genetic code, because only if you have understood what is genetic code, you'll be able to follow translation. George Gamow argued that the code should be a combination of bases, as there are only 4 bases to code for 20 amino acids. The process of replication and transcription, as we have already learned, it's based on complementarity. Based on the complementarity of these nitrogen bases only, both these processes are taking place. Whether it is replication or transcription, both are based on the rule of complementarity. The process of translation. So we already learned what is replication and transcription. In replication, genetic information from DNA is getting copied to DNA, whereas in transcription, the genetic information from DNA is getting copied to RNA. And translation is a transfer of genetic information from a polymer of nucleotides to a polymer of amino acids, or else you can say the polymer of nucleotides, that is RNA. RNA is a polymer of nucleotides. So the information present on this messenger RNA is getting converted, or the genetic information is being transferred to a polymer of amino acids. And you know, proteins are the polymers of amino acids. When the monomer units, amino acids, join together, proteins are formed. So polymer of nucleotide to polymer of amino acids means from messenger RNA. RNA and DNA both are polymer of nucleotides. So from the polymer of nucleotides, genetic information is getting transferred to amino acids, or it is getting translated into proteins. No complementarity exists between nucleotides and amino acids. They are having entirely different structure. So nucleotides, between nucleotides and amino acids, there is no complementarity. The change in the nucleic acids, the change in the genetic material is responsible for the change in the amino acids in protein. So when the nucleic acids, or when there are changes in the genetic material, accordingly there will be changes in the amino acids which are produced, even though there is no complementarity existing between nucleotides and amino acids. Depending upon the changes in the genetic material or nucleic acid, or depending upon the changes happening in this RNA, the amino acids can also change. there must be a genetic code. You should have a genetic code so as to direct the sequence of amino acids in proteins during translation. You should know what sequence of amino acids. There are different 20 amino acids are there. So what combination of amino acids are forming proteins? So different proteins are also there. So what combination of amino acids will form a particular protein? So to guide that, there is a need for a genetic code. There must be a genetic code to direct the sequence of amino acids in proteins during translation. You will understand all these once we complete it. I'll repeat it again. Replication and transcription both are based on the rule of complementarity. So in replication, you know, the nucleic acid or the information is copied from the nucleic acid DNA to RNA. Whereas, sorry, in replication, DNA to DNA, and in transcription, DNA to RNA. Both cases, we learned that it is happening on the based on the rule of complementarity. Whereas in translation, the information present in the nucleotides, in the polymer of nucleotides, that is RNA or nucleic acid, is getting translated, or it is getting transferred to a polymer of amino acids or protein, or messenger RNA is getting translated into proteins. So there is no such rule of complementarity between nucleotides and amino acids, because amino acids are not having any nitrogen bases like this. And the change in the nucleic acid, but at the same time, if there are some changes in the genetic material that can bring about, if there is some change in the sequence of these nitrogen bases in the nucleic acid, that can definitely affect the sequence of amino acids or the amino acids in nucleic acids. And there must be a genetic code to direct the sequence of amino acids in proteins during translation. When translation process, because you know that each protein is different. The sequence of amino acids present in each protein is different. The sequence of amino acids will decide the nature of protein. So there should be a genetic code to direct the sequence, which all amino acids should join together, in what order they should join together to form a particular protein. For directing all that, there should be a genetic code. The scientist George Gamow, he argued that the genetic code that we were speaking about should be a combination of bases. The genetic code should be made up of different nitrogen bases. It should be a combination of nitrogen bases. And we know that there are only 4 nitrogen bases: adenine, thymine, guanine, cytosine. And the only difference is that thymine is replaced by uracil in the case of RNA. Only 4 nitrogen bases, but we need 20 amino acids. We need to code for different amino acids. 20 amino acids are there. So with this 4, we need to manage for 20 amino acids. So he is saying, or according to him, this genetic code comprises of nitrogen bases. And this genetic code should code for 20 amino acids. And we have only 4 nitrogen bases. So he suggested that in order to code for 20 amino acids, at least there should be 3 nucleotides. Minimum there should be 3 nucleotides to code for 20 amino acids. So we have only 4 nitrogen bases and we need to code for 20 amino acids. So he said that minimum 3 nucleotides or minimum 3 nitrogen bases should be there in the code so that it can code for 20 amino acids, because the code for different amino acids are different. It's not the same. So here on the right hand side, you can see AAA, G, AG, UAU. So each one will be coding for a different amino acids. So to code for 20 amino acids, we have only 4 nitrogen bases. And George Gamow suggested that there should be at least 3 nitrogen bases so that it can code for 20 amino acids. A permutation combination. Permutation combination means by rearranging the order. like GAG, then GUG, like that, by rearranging it, by rearranging the existing nitrogen bases. A permutation combination of 4 raised to 3. It is not 43 children, it is 4 raised to 3. Just a minute. A permutation combination of 4 into 4 into 4 or 4 raised to 3, because we have 4 nitrogen bases, and in each code, as per George Gamow, there should be minimum 3. In each code, there should be 3 nitrogen bases, and we have only 4. So the maximum possible combinations are 64, 4 raised to 3, 4 into 4 into 4. So maximum we'll get 64 codones. This is more than what is required. We need to code only for 20 amino acids, but by a permutation combination, we will be getting maximum 64 codones. Are you getting it, children? Are you following?
Could you explain once?
Okay, I'll repeat it. For translation process to proceed, you know that in translation, the genetic information present on the nucleotide is converted to, or it is transferred to protein. You know, proteins means a sequence of amino acids, and between nucleotide and amino acids, there is no complementarity. So we need a genetic code to direct the process of translation. So scientist George Gamow suggested that for directing this translation process, for carrying out translation process, a genetic code is required. And according to him, this genetic code should consist of nitrogen bases, and only 4 nitrogen bases are there, and amino acids are 20 in number. So with this 4 nitrogen bases, we have to code for 20 amino acids. And he also suggested that each code can have minimum 3 nitrogen bases. Even though 4 are there, 3 is enough. 3 are enough to code for a particular amino acid. And if we are rearranging the existing 4 nitrogen bases, if you rearrange in different order, with each code having just 3, 4 raised to 3, we'll get 4 into 4 into 4. Maximum possible combinations we get is 64. We'll be getting 64 combinations of the nitrogen base. I'm repeating again, we have only 4. And each code need to have only 3 nitrogen bases. When you rearrange, so here you can see ACA, CCC, CG. So different ways, maximum possible combinations available are 64. And with the 64, it is more than enough for coding for 20 amino acids. Hargovind Khurana, he developed a chemical method that also helped in synthesizing RNA molecules with a desired combination of bases. So details of all these you don't have to learn, but what was their contribution, you should know. I'm repeating again, the scientists, all the scientists' names and their contributions are important. So Hargovind Khurana, he developed a chemical method, and with this method we were able to, like, desired type of proteins we are able to synthesize. His chemical method helped in synthesizing RNA molecules with the desired combination of bases. So artificially we can synthesize RNA molecules with the desired, what combination of bases we desire to have. In such a pattern you can prepare RNA. So then if the combination of bases is according to what we like, once it gets translated into proteins, the proteins also will be of our choice. So that was his contribution. And homopolymer and copolymer, the difference is that in homopolymer, all the monomer units, you know that the monomer units join together to form a polymer. If you take the case of protein is a polymer, monomer units are amino acids. Homopolymer means monomer units will be identical, same identical monomer units. And copolymer means when the monomer units are different, 2 or more kinds of monomers are joined together to form a polymer, you call it as copolymer. Just the opposite. Homopolymer, monomer units will be identical, will be same. Copolymer, monomer units will not be the same. There can be 2 or more kinds of monomer units. So what was his contribution? Har Govind Khurana, he developed a chemical method. And with the help of this method, they were able to synthesize RNA with desired sequences. You know that messenger RNA means it's a nucleotide. It is having a sequence of nitrogen bases. So if the nitrogen bases or the sequence of nitrogen bases, we can decide, definitely we'll be able to decide the proteins form, because it is depending upon the nitrogen bases that amino acids are formed. And it is depending upon the amino acids, the sequence of amino acids, proteins are formed. Marshall Nierenberg, he suggested a cell-free system. Outside a living cell, he conducted experiments, and this was meant for carrying out protein synthesis. So another scientist, Marshall Nierenberg, he also carried out experiments to learn the synthesis of proteins outside a living cell, and that also helped in discovering genetic code. The main person behind was George Gamow. All they contributed, all these scientists too contributed for the same. Severo-Ochoa enzyme, that is, the enzyme is named after the scientist Severo-Ochoa, and the name of the enzyme is polynucleotide phosphorylase. This enzyme was also helpful in polymerizing RNA with the desired sequences in a template-independent manner. In all these 3 cases, or all these 3 scientists, they decided to manipulate the synthesis of proteins. Usually, it is happening naturally in our body. Naturally, this process is taking place. The information present on RNA is getting translated into proteins. But these scientists worked out to find out whether they can synthesize messenger RNA chemical method, or by trying out in cell-free systems, and with the help of the enzyme and all, they were able to decide the order of bases in messenger RNA synthesized. So when the bases they can alter, they can even decide the type of proteins or the sequence of amino acids, and thereby the protein synthesized can be decided. So details of all that you don't have to learn, but you should know what was the contribution of each scientist. So I'll sum up towards the end. It finally gave rise to the checkerboard for genetic code. So all their contributions together gave the checkerboard for genetic code. You will see the checkerboard over here. You can see the checkerboard here. See here, they have written all the different nitrogen bases, T, C, A, G. Left hand side also you can see, and different combination. Maximum, you can see 64 different combinations. 64 combinations you can find over here. (64 combinations of nitrogen bases you can find here. See, T, T, T. See, 3T. This T, this T, this T, then again T, T, C. So the maximum, with this checkerboard, they tried out maximum possible combinations, and they got 64 combinations. And children, here you can see, hope you can see it clearly. Can you see the figure clearly? Yes, okay. Now, here you can find TTC. I told we have just 20 amino acids, but we have 64 codons. 64 codons are present, and three of them do not code for anything, and balance we have 61. I'll repeat again. We need to code only for 20 amino acids, and we have 64 codons. Each codon is a sequence of 3 nitrogen bases, and out of this 64, 3 are stop codons, so they don't code for anything. So balance 61. So we have 61 codons to code for 20 amino acids. And here you can see each amino acid is coded by more than one genetic code. See, abbreviation of all these amino acids are given here. ALA means alanine, arginine, aspartic acid. So abbreviations are given here for each amino acids. Now see the checkerboard, children. TTC. TTC means 3-thymine, thymine, thymine. Okay, TTC, thymine, thymine, and again cytosine. So these two are coding for Phe. Phe means phenylalanine. So those two codons will code for the amino acid phenylalanine. Similarly, TTA, TTG, they code for leucine. Similarly, you can find that from this, what you understand is for each amino acid, not just one codon. There are several codons. See here, you can see for threonine, 4 are there. 4 codons are there for threonine. So for each amino acid, more than one codon is present. Okay, more than one codons are there for each amino acid, and some of them just one. Methionine, just one. Okay, similarly, for others, 2 are there. For some, 2 codons, for some 4 codons. So that differ. What you need to understand is there are more than 1 codons for amino acids. It is not limited to just one, because we have 61 and only 20 amino acids. So each amino acids can have more than 1 codons. The codon is a triplet. Means 3 nitrogen bases. It has got 3 nitrogen bases. 3 nitrogen base sequences constitute 1 codon. There are 64 codons. There are 64 codons. 61 code for amino acids, and 3 of them you call as stop codons. You should know which are the stop codons. Usually you don't have to learn all these sequences, all the genetic codons you don't have to learn, but some of them you will have to learn. For example, stop codons you should know, start codon, that sequence you should know. Similarly, which are the different stop codons, all that you should know. Here you can see, this is one. Here you can, I have underlined the 3 stop codons over there. One codon, these are the features of genetic code. Codon is a triplet. First point, it's a triplet. We have triplet codons, and this triplet is formed by 3 nitrogen bases. And there are total 64 codons, out of which 3 are stop codons. They do not code for any amino acid, whereas 61 will code for different amino acids. Second point, one codon codes for only amino acid. Hence, it is unambiguous and specific. It's not vague, or it is, we have clarity in that, because one codon is coding for only one amino acid. Means, take the case of GTG. This will code only for valine. Or GCG, it will code only for alanine, not for any other amino acid. So you can say it is unambiguous and specific. A particular codon will code for only one amino acid. Unambiguous means precise, clearly expressed. There is no room for any doubt, because it's clear. This codon means that particular amino acid. Next. Just a minute. Then, third point, some amino acids are coded by more than one codon. So you can say it is degenerate. So you should not have confusion with the second and third statement. Second statement means a particular codon. A particular codon will code only for that amino acid. So GTG, it will code only for valine. It will not code for alanine, arginine, or aspartic acid. That is specific. But at the same time, Same amino acid can have different codons, maybe 4 codons. Here you can see valine is having 4 codons. All these 4 codons will code only for valine, not for any other amino acid. So don't get confused. The second point is that a particular codon is specific for a particular amino acid. We call it as unambiguous. Unambiguous means very precise and specific. It doesn't code. A particular codon is only for a particular amino acid, not for 2 different amino acids are not having the same codon. A particular codon is only for a particular amino acid. They are specific. And the third point is that same amino acid can have more than 1 codons, and that condition you call as degenerate. The codons are degenerate, means some of the amino acids are coded by more than 1 codon. Then the codon is read in messenger RNA in a contiguous fashion. Here this N is not there. Okay. And it is read in a contiguous fashion means it is read continuously, adjoining. These are very adjacent, no punctuations in between. We read it in a continuous way. These are lying very close to each other. There are no separations, no punctuations. That is read in a continuous manner. Then the code is nearly universal. The code is universal except in mitochondrial codons and certain protozoans. Means all the living organisms, the codon, the same codon codes for that particular amino acid, except in certain mitochondria and also in certain protozoans, differences can be there. In all other organisms, the same codon codes for a particular amino acid. For example, UUU, it is coding for phenylalanine. UUU, and in the picture given, children, it is shown as T. Wherever T in messenger RNA, T should be read as U. Okay, UUU will code for phenylalanine. In all organisms, from bacteria, bacteria is a prokaryote, to human beings. So that is why it is said it is universal. The codons are universal. The same codon codes for a particular amino acid from bacteria to the higher organisms like human beings. But there can be some exceptional cases in the case of mitochondria and also in certain protozoans. Now, AUG. AUG has got dual function, a repeatedly asked question. What are the dual functions performed by the codon AUG? AUG, it codes for the amino acid methionine, MET, abbreviated form. It codes for methionine, and at the same time, it starts as the initiator codon. Means the translation process begins from this particular codon. AUG, it acts as the initiator codon where the process of translation begins, because this particular start codon will be recognized and the process will be starting from AUG. So it is a start codon and it also codes for the amino acid methionine. Non-overlapping. Genetic code is read linearly. There is no overlapping. So each codon is separate and they are non-overlapping. Direction. The code is always read in 5' to 3' direction. Then anticodon. Transfer RNA has got this anticodon. Each codon is having a complementary anticodon on transfer RNA. Anticodon means, children, say for example, if AUG is present on messenger RNA, the anticodon present on transfer RNA will be, what will be the codon present in the anticodon? UAC. Understood? So for each codon on messenger RNA, transfer RNA will be having an anticodon. And why this anticodon and why this transfer RNA is needed, we will learn. So each codon has got a complementary anticodon on transfer RNA. Then nonsense codon, same as stop codon, UAA, UAA, GUA, UAG. They do not code for any amino acid. Okay, UAA, nonsense codons are those codons which do not code for any amino acid. UAA, UGA, UAG. You should know it by heart. You don't have to learn the entire codons, but these, some of them like start codons, stop codons, you should know they do not code for any amino acid, and there are no anticodons on transfer RNA for these three codons. Example, if the sequence of nucleotide in messenger RNA. See here they have given the example of how the genetic codons are getting converted to or getting translated into sequence of amino acids. See, say for example on messenger RNA, these are the sequences: AUG, UUU, UUC, UUC, UUU, UUU, UUC. And the sequence of amino acids, you know, AUG, start codon. You can refer either the figure given on the board, but here you have to replace all thymine by uracil. So AUG is methionine, UUU phenylalanine. You can check with your textbook. The table given there, the checkerboard given there, you can check. And UUC again phenylalanine. UUU also codes for phenylalanine. So phenylalanine and UUC again. So methionine, phenylalanine, phenylalanine, phenylalanine. So many phenylalanine because it is having so many codons. Several codons are there which will code for phenylalanine. UUU and UUC, both of them. UUU and UUC both codes for, or both of these codons code for phenylalanine. This is how it is getting decoded, and depending upon the sequence of amino acids, the proteins will be formed. Is it clear? Is it clear, children? Have you understood what is meant by a genetic code? Yes or no?
Ma'am,
Yes,
Yes.
you explain the anticodon part.
Anticodon. Okay, I will tell you. See, I told you that in translation process, the information present on messenger RNA will be translated into proteins or will be translated into a polymer of amino acids. Okay, and messenger RNA will be having so many codons. I told that each set of 3 nitrogen bases will form a codon. So proportionate to that, matching to that, there will be an anticodon on transfer RNA, because these transfer RNA are the one which will carry amino acids. They will read the code present on messenger RNA. Suppose it is AUG. So the transfer RNA carrying, so you know AUG codes for methionine. So the transfer RNA having methionine will reach there and they will read the sequence where it is AUG. And then on this transfer RNA carrying methionine, they will have the anticodon for AUG. Like AUG means what will be the anticodon? UAC.
See.
So that will be present on transfer RNA. So they will come and recognize. They will find out where is the matching codon. Transfer RNA with anticodon will reach the site and they will check where is the matching codon. And there they will transfer the amino acids, which you will understand when we learn translation. Okay, Punya? So that process you will understand what is the role of transfer RNA and what is the significance of anticodon present there, you will learn. Then tell me the features of genetic codon. What are the different features that we discussed? Rijas, 1 point. Louder.
They are in triplet.
They are triplet or these code. Triplet means what makes this triplet?
3 bases.
Three
Nitrogen base.
Three nitrogen bases. Total, how many codons are there, Evelyn?
64.
How many are functional
61.
and remaining?
Remaining 3 are non-functional.
They are stop codons or nonsense codons. Nandana, another point.
One codon codes for only one amino acid.
One codon codes only for one amino acid. Lydia, another point.
Ma'am, I didn't write that.
Louder.
I didn't write that.
Any point you can say. I told so many points, no, Lydia. Any one point. Check your textbook and let me know. Michael.
More than one codon, so they're degenerate.
They are degenerate. What do you mean by degenerate?
I mean, they're not functional.
Some amino acids are coded by more than one codon. Meenakshi, next one.
Always read in the direction from 5 prime to 3 prime.
It is read from or read in 5 prime to 3 prime direction. So we'll continue next class. Okay, thank you, children. See you next class.